In terms of background, I want to briefly mention 4 things: CRISPR, its associated protein CPF1, the dystrophin gene DMD, and Duchenne muscular dystrophy, the disease that results from dysfunctional dystrophin. CRISPR stands for clustered, regularly interspaced palindromic repeats. In nature, CRISPR and its associated proteins cut out DNA from invading viruses and store them in a bacterial genome as spacers between palindromic repeats, as shown. The harvested viral DNA can later be transcribed into guide RNAs that help CRISPR-associated endonucleases hunt down other instances of the viral DNA via base pairing, and then introduce double-stranded cuts that result in the degradation of the viral DNA. One such endonuclease, CPF1, was used in these experiments instead of the first discovered CRISPR-associated endonuclease, Cas9. This was done for a few reasons. The most important of these reasons is that every CRISPR-associated endonuclease has its own sequence of nucleotides, called PAM, that it has to find near its target sequence before it cuts. This prevents the endonucleases from cutting up their own bacteria's CRISPR array. CPF1 happens to have a PAM that exists at the intron-exon boundary before exon 51, which is exactly where the experimenters are directing CPF1. DMD, the gene that encodes for dystrophin, is the largest known human gene. It is found on the X chromosome and has 79 exons and over 2 million base pairs. And it is expressed mainly in muscle cells. The protein is effectively a large rod which attaches to the intracellular cytoskeleton on one end and to a transmembrane protein on the other end. Non-functional protein can result in Duchenne muscular dystrophy, which causes debilitating muscle atrophy over time and will eventually become respiratory or cardiac failure. In the following experiments, researchers attempt to rescue DMD mutant cells with CPF1 via 2 different strategies: exon skipping and introducing insertions and deletions.
Another method used by scientists to restore dystrophin expression was termed exon skipping. You can see in figure A that the G2 or G3 guide RNA guides the LBCPF1 endonuclease to intron 50, while the G1 guide RNA guides the endonuclease to exon 51. The G2 guide RNA had higher cleavage efficiency and was therefore used for the purposes of this experiment. Figure B shows the results of an assay where the red arrowheads denote the cleavage at intron 50, while figure C, the lower band is marked with the red arrowhead and is the fragment that corresponds to the removal of the exon 51 splice acceptor site. Figure D sequences this fragment shown in figure C, and it shows a 200 base pair deletion of the exon 51 AG splice acceptor site, which is the predicted result based on these tests. Figure E shows the results of another PCR experiment. The fragments are labeled. The lower band shows the skipping of exon 51. Since this exon was skipped, less base pairs were being read. Therefore, this fragment was smaller and it was able to migrate farther through the gel. It's labeled at the bottom. Figure F sequenced this fragment, and the DMD gene was actually reframed, and the stop codon at the end of exon 51 was actually bypassed, allowing for dystrophin expression to be restored. Figure G is a Western blot experiment. The band labeled dystrophin was consistent with the structural, with just the structural formation or the structure of dystrophin, and this therefore confirmed that dystrophin was now being expressed. And figure H shows immunocytochemistry results where the red staining shows that dystrophin expression had been restored. Uncorrected cells did not show any red staining. Dystrophin expression was not occurring, but after correction with the LBCPF1 endonuclease exon skipping method, you see the red staining showing that dystrophin expression had been restored.
In addition to exon skipping, there's another method called indels. This stands for small insertions and deletions, and obviously it refers to the insertion and or deletion of nucleotides into genomic DNA. This event must be less than 1,000 base pairs in length, and it also will change the overall number in the DNA. An indel must be a multiple of 3, otherwise it will result in a frameshift mutation due to the triplet nature. So next we have the first figure of the experiment, and this shows the correction of DMD mutations by CPF1 mediated genome editing. So in part A, you'll see the 2 methods for the deletion of exons 48 to 50, either by reframing using indels or the exon skipping method as discussed. And in part B, you'll see the 3' end of the intron is T-rich, generating CPF1 PAM sequences, which enables genome cleavaging. And then you'll see in part C that CPF1 targets DMD exon 51, and these red arrows show the staggered end distal to the PAM site. Next, in part D, you'll see the plasmid encoded with human codon optimized CPF1. Also, it is encoded with NLS, which is a nuclear localization signal, which essentially tags a protein for import into the nucleus via nuclear transport. It's also encoded with 2A GFP, which is green fluorescent protein, and chicken beta-actin promoters, labeled as CBH. Cells transfected with this plasmid express GFP, which allows for selection of CPF1 expressing cells via fluorescence activated cell sorting. And then lastly, in figure E, you'll see the assay comparing human 293 T cells versus the stem cells, the DMD stem cells transfected with the plasmid, expressing either LBCPF1 or ASCPF1. These are CPF1 orthologs, and an ortholog is genes in different species that have evolved from a common ancestral gene, but they retain the same function. So, and then the red arrows denote the cleavage products, and then you'll see the marker under M and then base pair length.
Figure 2 is a collection of 9 varied subfigures that show the methodology as well as the results of the correction by indel mutations. Figure A shows just what the experiment was and shows the differentiation of the iPSCs into cardiomyocytes. And figure B shows the exons that are being skipped, as well as shows the binding sites for the reverse transcriptase PCR primers that verify the identity of the corrected transcript. Figure C shows subfigure C shows the intention of the experiment, with after the premature stop codon shown in the uncorrected being removed in the reframed one. And this prevents the truncation of the protein, blocking the dystrophy from manifesting in some cases. Then the next 4 subfigures are just results slides. Figures D and F are Western blot analyses showing the restoration of dystrophin, as shown by in figure D, the thick bands around 250 kilodaltons, and in figure F, the narrow bands also at 250 kilodaltons. As noted, figure D is polyclonal, suggesting that there's various different splices of dystrophin, which is why you see those blurry bands. Whereas figure F, both of those results are monoclonal for clones 2 and 5, which were successful, but of different sizes. Figures E and G are immunocytochemistry, and they are intended to show the restoration of dystrophin, with the red dye showing where dystrophin has been restored, and as shown, it's not present in the uncorrected cardiomyocytes, but it is present in the wild type and those treated by the CPF1 splicing mechanism. Subfigures H and I are 2 graphs that are intended to compare the wild type and the corrected cardiomyocytes. Figure H shows the mitochondrial DNA copies, and there's no statistical difference between the wild type and the corrected clones, suggesting that the dystrophin and truncation is no longer, at least if not present, it's no longer negatively impacting the mitochondria of the cell. While figure I shows the oxygen consumption rate under various inhibitors and uncouplers, and the results are a little bit more varied here, with some aspects of respiration happening in decreased rates in the corrected relative to the wild type, particularly under the influence of FCCP, which is an uncoupler, which boosts oxygen consumption. We do not see the same behavior between the wild type and the corrected cardiomyocytes.
Scientists now wanted to use a CPF1 mediated mechanism to correct dystrophin expression in MDX mice. Specifically, they were focused on the nonsense mutation, exon 23 of the DMD gene, shown in figure A, where a C to T transition causes the formation of a premature stop codon, preventing the expression of dystrophin. LB-CPF1 and ASCPF1 are two different orthologs of the CPF1 endonuclease, which cleaves DNA at a specific site. They have different cleavage efficiencies, shown in figure C. Additionally, different guide RNAs target different exon 23 regions, this is shown in figure B, and through the results of these tests, it was determined that LB-CPF1 used with the G2 guide RNA generated the greatest occurrence of indels in mouse fibroblasts, and therefore they were used for the purposes of this experiment. The specific technique used to correct the nonsense mutation exon 23 of the DMD gene is called HDR, homology-directed repair. Homology-directed repair utilizes a double-stranded DNA to break a DNA strand and then uses a template to repair this cut DNA. But there must be high sequence homology between the intact and cut DNA. For this experiment, the LB-CPF1 and G2 guide RNA made it cut 8 base pairs downstream of the mutation site after recognizing a PAM sequence 9 base pairs upstream of the mutation site. A 180 base pair SSODN was used for this experiment as the HDR template with LB-CPF1 and G2 to allow for gene editing. 90 base pair sequence homology flanking the cleavage site was seen in the SSODN HDR template, making it ideal for correcting the nonsense mutation exon 23 of the DMD gene. After completing the HDR mediated reaction or mechanism, they introduced 4 silent mutation in the TSE1 restriction site that allowed for genotyping, and the high sequence similarity between the HDR template and the cut DNA strand allowed for repair, and this repair was found to restore dystrophin expression, confirming that a CPF1 mediated mechanism can effectively correct dystrophin expression in MDX mice.
Correction of muscular dystrophy in MDX mice. LB-CPF1 mRNA, G2 gRNA, and an SSODN template containing a restriction site were co-injected into MDX mice zygotes, which were then reimplanted into a pseudopregnant female. Zygotes were corrected with either non-homologous end joining or homology-directed repair. 12 of the 24 mice seen with this T7E1 assay were found to have been edited. Of those 12, only 5 carried the restriction site that indicated HDR correction, seen with this RFLP assay. These 5 were designated MDXC mice, which had their stop codon deleted. Histological staining with hematoxylin and eosin of some muscles showed MDXC mice had reduced inflammation and fibrosis. Immunohistochemistry with dystrophin antibodies showed MDXC mice had corrected dystrophin expression, indicated with red, similar to wild type. Creatine kinase is released into the blood when there is muscle damage. Therefore, elevated CK levels are indicative biomarkers in muscular dystrophy. MDXC mice had decreased serum concentrations of CK compared to MDX mice. This was inversely correlated with the percentage of correction. A 4-limb grip test showed that corrected mice had rescued muscle strength, indicative of functional correction of muscular dystrophy.
So in conclusion, for the entire experiment, there were 2 strategies used, reframing and exon skipping. They were applied to restore the open reading frame of the DMD gene. Both strategies rescued metabolic abnormalities found in DMD stem cell-derived cardiomyocytes. LB-CPF1 mediated HDR correction rescued pathophysiological hallmarks of the dystrophin gene in mice. And then these are some outstanding questions.